Association, Mr. Vice Minister of Agriculture and Rural Development of Albania, Directors of the Medical University of Tirana, of the University Alexander Giovanni of Elbasan, Dean of Faculty of Natural Sciences of the University of Tirana, dear Rikrop family and Dr. Franco Stefanakis, dear EBTNA family, welcome to Tirana. In the first ever EBTNA Congress to be held in our country. I'm very happy to have you all here, and on behalf of the organizing commission, to declare this Congress open. So on this occasion, allow me to thank Professor Dundar, who is not present here today, and the EBTNA Executive Committee members who made possible the organization of this event and brought all of you here. The same thanks go to the President of the Academy of Sciences of Albania for supporting and hosting this Congress and giving us the possibility to have all of you here in this venue, as well as for supporting 2 out of 4 satellite events to this Congress. Also, many thanks go to Dr. Matteo Bertelli, who is here with us as the president of the MAGI group for the rare genetic diseases, who also has organized a special session to the Congress and a international workshop, hands-on medical genetics. Many thanks go to Dr. Stefanakis, the chair of the Rikrop Cost Action, who is organizing a satellite workshop on plant resilience in the conditions of climate changes, as well as to Professor Robert Marks from Ben Gurion University of Negev, who organized 2 of 4 satellite events to this Congress. Thank you very much to all of you. Now, please allow me to give the floor to the President of the Academy of Sciences, Akademisë Shqipërisë, for the greetings of the Congress. You want? Okay, so together with the Vice President of the EBTNA, Dr. Oscar Vicente Mana, we may sign first the agreement between EBTNA and the Academy of Sciences of Albania, and then we may proceed with the greetings. Yes.
Përshëndetje, mirë se keni ardhur. Pra, sot kemi kënaqësinë ashtu si për të hapur këtë Kongres Evropian për bioteknologjinë që zhvillohet në Tiranë, firmosim dhe një marrëveshje me EBTNA-n, me cilën tashmë ne kemi një traditë të gjatë bashkëpunimi. Pra, firmosja është një akt formal, sepse prej 8 vjetësh bashkëpunimi midis Akademisë Shkencave, njësive të saja të reja të kërkim studimit dhe universiteteve, Universitetit Tiranës, Universitetit Mjekësisë, Bujqësor, me radhë, ka ardhur gjithmonë duke u forcuar veçanërisht në fushën e bioteknologjisë. Kështu që nisur nga kjo përvojë, ku janë organizuar një sër konferencash, disa shkolla me studentët e rinj, me doktorantët e rinj, ashtu si dhe ndërmarrja disa projekte të përbashkëta, ka çuar në atë që në mënyrë të natyrshme të firmosin këtë marrëveshje së bashku me profesor Oscar, dhe duke shprehur dhe një herë vullnetin, dëshirën e Akademisë Shkencave për ta çuar më përpara këtë bashkëpunim. Dhe organizimi i këtij kongresi është pikërisht produkt i këtij bashkëpunimi. Ne jemi shumë të nderuar që presim nga profesorët dhe kërkuesit më eminent në fushën e bioteknologjisë këtu në Tiranë, pikërisht dhe në vitin ku Tirana është shpallur kryeqytet i kulturës evropiane, kështu që duhet kombinuar kultura me shkencën dhe organizimi i këtij kongresi tregon dhe atë që njësitë tona kanë forcuar shumë bashkëpunimin me shkollat e larta. Akademia e Shkencave e vlerëson këtë bashkëpunim, e shet të domosdoshëm, pavarësisht që ndonjëherë jo gjithë universitetet e shohin të domosdoshëm bashkëpunimin me Akademinë e Shkencave. Por me këtë rast përshëndes dhe prezencën dhe të rektorit të Universitetit të Tiranës, dekanës Fakultetit Shkenca e Natyrës, përfaqësuesit të universiteteve dhe të profesoratit të nderuar të tyre. Ashtu siç njëna qëllimet e këtyre aktiviteteve është dhe sensibilizimi i politikës për të zbatuar arritjet e reja, veçanërisht fushën e bioteknologjisë. Kështu i përshëndes me këtë rast dhe prezencën e zëvendës ministrit të bujqësisë, duke dëshiruar një bashkëpunim gjithnjë e më të frutshëm. Kongresi është i shtrirë në tëra disiplinat e nëndisiplinat e bioteknologjisë, dhe mund mbetet vetëm të uroj punime të mbara, falenderoj profesor Dundar që ka drejtuar me shumë sukses këtë asociacion Rrjet të Bioteknologjisë Europiane, të përshëndes organizatorët dhe të uroj shumë sukses Kongresit Europian të Bioteknologjisë që organizohet në mjediset e Akademisë Shkencave, dhe që është një nder për ne që 3 ditë me radhë do të jemi të mbytur në bioteknologët. Suksese!
Thank you very much, Mr. President.
So, good morning to everybody. On behalf of the European Biotechnology Thematic Network Association, EBTNA, I am very happy to welcome you here to our Congress. Yeah, the seat of our association was found more than 20 years ago in Perugia, in Italy, and I would like to remember our founder, Professor Maria Pia Viola Magni. We started with the organization of international first-level degree on biotechnology, job-oriented biotechnology, with a consortium of different European universities sending students to Perugia. And this was a bit the origin of this association. We had been organizing this European Biotechnology Congress for more than 15 years. All over Europe, we have been in Italy and Spain, Slovakia, Slovenia, Romania, and now for the first time in Albania. I am very happy that we are here. I would like also to thank Ariola and her team for the organization, the Academy of Science, of course, for providing the venue and support, and also people involved in our Congress and all satellite events, the cost meeting, the workshops. And so I think we have ahead 3 days, 3 days of science, of networking, that I really, I am sure will be really very useful for all of you. And I hope also you will have some time to enjoy the city. And since we are here mostly for science, not to listen to speeches. I think I will cut mine short. And again, thank you very much and welcome to Tirana for the European Biotechnology Congress. Thank you.
Thank you, Professor. Thank you very much, Mr. President of the Academy of Sciences and Vice President of EBTNA. We proceed with the next greeting. So I have the pleasure to invite the Rector of the Medical University of Tirana, Professor Draccini, to greet the Congress.
Honorable professors and colleagues, distinguished representative of academic and scientific institutions, dear participants and friends. It is a special pleasure and a great honor for me to be among you today at the opening of the European Biotechnology Congress 2025, which this year is being held in Tirana. This is an important moment, not only for the international scientific community, but also for Albania, which has the privilege of hosting such a significant and prestigious event. The organization of this Congress by the European Biotechnology Thematic Networks Association is a clear evidence of the growing role that our country is assuming in the map of European and international scientific cooperation. This interinstitutional collaboration, grounded in knowledge and research, stand as a guarantee for the advancement of science and for the strengthening of cooperation between our academic communities. This year's Congress, with its topic ranging from molecular biotechnology, genetic therapies, environmental, industrial, and food biotechnology, is a direct call for collaboration and for finding scientific solutions to global challenges of our time, confronting new diseases, ensuring healthy and sustainable food, protecting biodiversity, and responding to climate change. It is in particular a great privilege for me to open the MAGI session dedicated to supporting the new generation in the fields of biotechnology. Young scientists are not only our successors, but also the leaders of tomorrow. By investing in their education and development, we safeguard the future of sciences, ensuring continuity, innovation, and social responsibility in the application of biotechnology for the benefits of the humanity. I wish to express my deep gratitude to Professor Muniz Dundar, President of EBTNA, for his vision and commitment to scientific cooperation. Special thanks also to Professor Tommaso Beccari for his academic contribution and to Professor Natalia Capodicaza, who has played a vital role in building bridges coordinating this important event. Of course, a special thanks for Academy of Science for hosting such a prestigious event. I am convinced that the coming days will offer a valuable opportunity to exchange ideas, to strengthen existing collaboration, and to establish new partnership. The outcomes of this Congress will extend well beyond the walls of this hall, directly contributing to advancement of scientific research, to innovation policies, and to the improvement of the human life. Maybe this Congress stands as a concrete example of how science, when carried out with passion, collaboration, and visions, has the power to transcend borders and to unite cultures, nations, and generations. I thank you, everybody, for your presence and wish to everybody a fruitful and successful proceedings at this prestigious Congress. Welcome to Tirana, our friends. Thank you.
Thank you very much, Mr. Rector. And now we proceed with a greeting from the Dean of the Faculty of Natural Sciences of the University of Tirana, Professor Eglantina Kallucci. Welcome.
Good morning, everyone. Dear guests, dear colleagues coming from all around Europe, dear colleagues coming from diaspora. I am honored to be part of this activity and to greet this Congress. President of Academy of Science, thank you very much for the collaboration and continuous, in fact, collaboration that we have already established, exchanging our scientific contributions in various fields. So I would like to thank Professor Ariola Batu from the University of Tirana, and so lecturers from University of Tetovo, University of Pristina, and all the colleagues coming from all around, from Italy, Israel, and all around Europe and the world, from organizing this opportunity. In this welcoming ceremony, it is a great pleasure and honor for me to greet in the name of the Faculty of Natural Sciences and University of Tirana. At first, I would like to dedicate a special acknowledgement to the persons who contributed in the organization of this event, because since the idea came until today, it's a very large gap. So I would like to thank the faithful friends that trusted in us and had the idea of organizing the edition of the European Congress of Biotechnology here in Tirana. We evaluate it as a multidisciplinary contribution because it brings together a scientific network and makes this network larger and larger. This Congress contributes not only in giving knowledge, but offering young researchers, graduate students, doctoral students, and professionals from Albania and the region a broad overview how the development of technology and artificial intelligence has driven the process of biotechnology and has impacted the related disciplines in medicine, pharmaceutical production, human and non-human microbiome, and genetic data. The lectures and the discussions that will be held in this activity will not be beneficial only for the students and for the PhD and professional scientists, but also for our departments and our academic staff, leading to a direct contribution into the improvement of our program studies and our curricula and the bachelor and master programs. Something no less important is the opportunity of creating new communications between academics, upcoming joint projects, which goes in coherence with the actual Albanian higher education reforms towards internationalization. A special gratitude goes to the organizers of this activity, the colleagues of the Department of Biotechnology in the Faculty of Natural Sciences, University of Tirana, and all the contributors that in the most cases the work is not visible, but without their involvement we could not organize such a successful event. And I wish this will be one of them. The main goal of the event is to provide an opportunity for researchers from all around the world to present their research result and to discuss the practical challenges encountered. In fact, this activity will provide opportunities for these participants to exchange new ideas and application experiences and to establish research and to find potential partners for future scientific activities. In fact, I would like to share with you some personal remarks about this collaboration. In fact, I joined MAGI group at November 2019. And since then, in fact, we have established a very good and strong collaboration because my background is mathematician, but in fact, science does not have boundaries. So we could meet and find points of encounters and with exchange ideas, no matter our directions and our expertise is. So here I am glad to be part of the session that Maggie Group is going to organize, and also to share with you that we have exchanged and we are working together with common PhD students from Italy and Albania. And at the end, once again, I would like to wish a great success to the European Congress of Biotechnology 2025, and we wish that this will be a good omen for further activities. Thank you very much.
Thank you very much, Eglantina. Your support is so much appreciated, and you know that we're working together every day. So now allow me to invite Professor Matteo Bertelli, President of MAGI Group for Rare Genetic Diseases, who is a very close collaborator of our group for a long time now, and who has provided so much to the organization of this event. Thank you very much.
Thank you. I want to thank first Professor Ariola Baciu to organize this splendid event, and all the EBTNA family. Really, EBTNA are not a common scientific society, but are a family in which all can contribute to grow. For example, in Albania, this is the third course that we organize, because we have also a laboratory there in Albania. And this is the third course that we organize for the young student, to practical course on biotechnology. And many of these students also receive some grants to come to visit our laboratory in Italy and start to do some in particular research. For example, from my group now are present two persons that I meet for the first time at this event, and now work in Italy in our group, and work half in the university and half in our institute. And really, I think that the future are open to many opportunities. I thank you a lot, Professor Eglantina Callucci, because thanks to Professor Callucci, we start to develop a project between mathematics and genetics, and we publish 3 articles, and we start to develop a mathematical model to analyze the effect of genetic polymorphism on the phenotype. And for my side, what I think, what I believe is that we need to open the frontiers and create a network thanks to EBTNA, to the EBTNA, that help all the university around the country to promote the exchange and grow and project together. Thank you.
Thank you very much, Professor Bertelli. Your support is precious for us. So allow me to invite now Professor Robert Marks, who's going to greet us in the name of a big group from Ben Gurion University of Negev, Israel, who's been collaborating with us for at least 6 last years in different projects and organizations. Thank you, Professor.
So, as Ariola said, we had a number of years to collaborate together. And although the first time I came to Albania, it was not thanks to Ariola, but for the Albanians who know her, is Professor Bino. At any rate, since I've been here the first time, I think 7 times I've been in Albania, I'm impressed how you grow and grow and grow. Very impressed. So I said to Ariola, are you sure you want to organize EBTNA? And she said, yeah, of course. Are you sure you want to organize EBTNA? At any rate, of course she did. And as she said, we've been together in some of events she's organized here in Albania. And of course, she's always very good at doing that. And of course, we can see she's done a great job here. And I'm glad to see all my peers from EBTNA. I'm glad to see all the honorary guests, the presidents, rectors, and many others. And I hope this event, which is the most important thing, I think, beyond the science that we produce and that we share in the literature at any rate, is the networking. I always find a lot of friends when I come here. It's always a pleasure to meet them again and again and again, and some new friends. And yes, we organized 2 workshops at the request of Ariola. I would like to thank my students, Fred Narcross and Gal Carmeli, who are here, who hosted the students and helped them and coached them. Not only them, there's an ex-student of mine here somewhere, Evgeny, who also came here. Now he's an independent scientist, and I'm glad to see him here. I don't know if Ariel Kushmao is here yet, but a colleague of mine from my department is here too. Interesting for the Albanians here, I have 2 Albanian students too, not only Materi. I also have students, very happy with them. Both of them this year graduated as a PhD students. One is coming back in a week to Albania, the other one staying in Israel. What can we do? Postdoc. And yeah, we were very happy with them. And so we sort of created an interesting ecosystem between Israel and Albania, and hopefully that will continue. And in the hotel where I stay, in the last 5 days, every single day, bus loads of Israelis are coming. So, okay, I thought I was in Albania, but at any rate, it became a very nice destination for tourism for Israelis. And yeah, great place. Thank you.
Thank you very much, Professor Marks. And to continue with the greeting session, which we already almost finished, I have to mention that a few details on the structure of the program of the Congress. The Congress is organized in 40 sessions, which span different areas of biotechnology, like the medical, pharmaceutical biotech, plant biotechnology, environmental and marine biotechnology, aquaculture and animal biotechnology, biocatalysis, biopolymers, and nanobiotechnology. Thus, furthermore, 4 satellite events enrich the Congress program. Precisely, an international workshop on biotechnology for climate-resilient agriculture and urgent solutions for food security of cost action recrop, a training school on fostering regional entrepreneurship in biotechnology, 1 workshop hands-on medical genetics, and another one on hands-on cytotoxicity testing. Now, as you go through the program, you'll see how many important topics are to be covered, and you'll have the possibility to exchange expertise with different colleagues from all around the world. And when I mean all around the world, I can inform you that the Congress will have about 200 participants coming from overseas, from Canada, United States of America, China, from Australia, from which I thank at most because of the efforts they did to be here with us today, as well as from different European countries, Western Balkan countries, and Middle East. So we have all the world in here, in Tirana, in this occasion. Many thanks to all of you for being here. Now allow me to consider the greeting session closed, and we proceed with the first session, which is named Albanian Panel on Biotechnology. Okay, so the first to be presented at the Albanian panel is Professor Dr. Anila Hoda, General Secretary of the Academy of Sciences of Albania. Will you allow me please to read the short bio on Anila. She's a biologist, a researcher, and academic lecturer, currently serving as the Scientific Secretary of the Academy of Sciences of Albania. She has a degree in biology and a decade of experience in genetics, biotechnology, and higher education leadership. She served as the head of the Department of Animal Science and deputy rector at the Agricultural University of Tirana, where she lectured and conducted research in livestock genetics and biotechnology. So her scientific work focuses on animal genetics, molecular biotechnology, and population genetics, with particular expertise in genetic diversity and polymorphism in livestock breeds, molecular markers for breeding and conservation, and the genetic improvement of production traits. She became a full member of the ACAA, the Academy of Sciences of Albania, in 2023, where she's played a central role in the coordinating interdisciplinary national and regional research projects and leading editorial teams for the Albanian Encyclopedia in the fields of veterinary and agricultural sciences, environmental sciences, and medicine. So, floor is yours, Professor.
Thank you, Professor Batsu, for introducing me, and thank you for the organization of such important event. So, indeed, my presentation is a general overview with a focus on the development and application of biotechnology in the livestock and aquaculture sector in these two countries, Albania and Kosovo, which are located in this part of Southeast Europe. During the presentation, I will try to cover very briefly these arguments that you see here, and I will stop a bit wider in the highlighted areas. So biotechnology is of a great importance for the livestock and aquaculture sectors because due to the advanced techniques and tools, it makes possible to enhance the productivity, to improve the animal health, to make a sustainable sector, to reduce the risk of animal diseases, and everything will help also to align both countries, Albania and Kosovo, with the European Green Deal agenda. All of this make biotechnology not only an option, but a strategy for the future of our countries. But why the focus is livestock and the aquaculture sector? Not only because of the importance that these two sectors has for the economy of the country, but they are the livelihood of the farmers and the local community whose life is based on these two sectors. In many cases, these are the main sources of income for this community and their food security. Nevertheless, of this importance, the sectors are facing a lot of challenges, such as low productivity and outdated practices, which threaten the long-term sustainability. Therefore, it is an urgent need to modernize the sector, and this possibility is offered only by biotechnology, who can offer the solution for this kind of transformation, for modernizing production, improving efficacy, and also for aligning with the EU agenda and legal framework. So I will give some general information about the biotechnology in livestock sector and aquaculture. And starting with the livestock sector, the methods or techniques that are applied in both countries are two of them. Regarding the reproductive biotechnology, which are artificial insemination and embryo transfer. Both countries have a long experience with artificial insemination, and in Albania starting from years 1950. After years 1990, the sector or the tool went up and down, but actually the artificial insemination is routinely applied to the cows with the main focus to improve the milk yielding in cows. The same situation is in Kosovo, where is a center that produces enough semen from high genetic married bulls with this aim to improve the milk yielding in cows. In Kosovo, it was made possible in 2015, the first embryo transfer techniques, thanks to a collaboration with Iowa State University of United States, where it was made possible that a calf from a high married cow was born through a surrogate mother in Kosovo. And the examples are running even nowadays. Another argument I want to stop a little bit is the focus that two countries are paying attention to the preservation of local genetic resources. Both countries have a very rich biodiversity. In Albania are at least 42 native breeds of all kinds of livestock, cows, sheep, goats, buffaloes, etc. In Kosovo, the number is fewer, but anyway, both countries have a very rich biodiversity. And the native breeds are of great importance because we know that they have a good quality product. They are adapted to the environmental, to the management conditions. But there is a challenge. Their production is lower compared with the imported or exotic breed. And we have also another challenge that the villages, at least in Albania, are depopulating. The youth doesn't like to stay anymore in village, but they prefer to come in the city or to go abroad. Therefore, the government is supporting the sector by subsidies or also by the international organization who support financially the sector. There are a lot of native breeds in both countries, but I will show some more details about the most important of them, or maybe regarding them that are in the status of endangered. One of these is Balusha sheep that you see here from Kosovo. Is a typical breed that the researchers has shown that it has some genes that offer a high level of immune response. But the number of individuals was decreasing rapidly, ending with about 300 animals. The universities, the government, the international organization took care to conserve this breed, not only for these values that I mentioned, but it is a native breed and it is also part of cultural heritage. More or less is the same situation with some cattle breeds such as Busha, which is a breed that is found not only in Kosovo, but also in Albania and in Republic of North Macedonia. Here is a picture of Busha cattle, and in both countries are making efforts to conserve this breed. In Albania, there are two breeds, Shkodrane, sheep breed, and another one, Lara Policid, that are considered an endangered status. And here in this region, in Korça, in southeast part of Albania, is a center for agricultural technology transfer who is caring to conserve these important breeds. Great attention is paid to the disease control in the livestock sector, and both countries have two state-governed institutions that are Institute of Food Safety and Veterinarian, and the same in Kosovo, who are of national reference, and by the use of modern techniques such as PCR, DNA sequencing, molecular testing, etc., they take care to detect the rapid outbreak and also for the vaccination and the prevention of diseases outbreak. Aquaculture is also of a great importance as a sector that is developing rapidly in both countries. There are no direct genetic engineering in this sector, but the focus is on broodstock management and hatchery improvement. Albania has a very diverse aquaculture because this quite rich with water resources, such as rivers, lakes, and sea. Meanwhile, Kosovo is a landlocked country, and so in Albania is developing trout farming and also farming of marine cage culture. And the production of aquaculture is increasing rapidly in both countries, but Kosovo has experience with the rain-brown trout farming. In both countries, there are hatcheries who take care for the production of fingerlings or fries in trout and carp. Here are some photos from the Center for Cultivation of Mediterranean mussel, which has a long history in Albania and is located in southern part of Albania in Butrinti Lagoon. And here are the fingerlings of Ohrid trout production and their release at the Ohrid lake. The famous fish species in Albania is Ohrid trout or Salmoletnica, which is an endemic species that is raised, or its habitat is Lake Ohrid, that is a cross-border lake between Albania and North Macedonia. These are some photos of Salmoletnica during our sampling expedition. And in Pogradec, in Lin, there is a center who produce fingerlings that are released every year in the lake in order to conserve this species, because it is overexploited or overfishing due to the consumer demands. Then we have also other hatcheries. Again, in Lin is a recent one of this year, and next year we will have the first fingerlings of carp. And another hatchery is here in Prespa Lake, which produce fingerlings for this Cyprinus carpio. And the fingerlings are released only on Prespa Lake. I mean, they do not mix the lakes. And here is the Butrinti Lagoon, where the mussel, Mediterranean mussel, is raised since many decades ago. The biotech tool that is applied in fisheries sector is just the reproduction, artificial reproduction of some exotic Chinese cyprinids. Of course, the government pay attention to the sectors. Both countries have the law, have convention ratified, have their strategic plan with the main focus conserving the genetic resources, animal genetic resources, and their sustainable use. And of course, are the respective ministries who are taking cares, and international organizations such as IPARD or World Bank, etc., who are supporting also financially the sectors. Of a great importance are the research institutions and education institutions that are universities. So in Albania is Agriculture University of Tirana and the Faculty of Natural Sciences, who offer study programs on the biotechnology area. And in Pristina, in Kosovo, is University of Pristina, who also offers study programs in the biotech field. I like to emphasize the contribution of Academy of Sciences of Albania in this area of the research. So recently in our academy, it was established the Research Center of Biotechnology and Genetics, which has a sector of animal biotechnology. Actually, are running two projects. One of them is with the focus of the biodiversity in native sheep breeds with the support of National Agency for Research and Innovation and Center of Technology Transfer in Korça. And another project is the estimation of diversity of carp from natural lakes. In Academy, there is a commission that is called of Biology, Agriculture, and Veterinarian that is composed by well-known researcher of this field, who are focused mainly on the hotspots of the sector, and they give, let's say, suggestion where to be focused, and also through the debates that is happening during conferences, workshops, or roundtables, Academy acts as a bridge, let's say, between science and policy making and decision making bodies such as government. One of the main focus of the research has been the estimation of genetic diversity of the native breeds, sheep, goats, cattle, and also of the ochre trout salmoletnica that is performed recently. All this research have been focused on the use of molecular markers, which is the future for us. So the future for both countries has three key pillars that are innovation, collaboration, and sustainability. So through genomic at scale, seed and broodstock, ISO accredited labs, digital traceability, and climate smart system, so everything will help to accelerate breeding programs in order to improve the animal, livestock and fish performance, to have a healthy and resilient animal population, to strengthen food safety, and to fulfill the consumer demands and their trust, to increase the export potential, to increase the transparency, and to be aligned with the EU regulation. And finally, to reduce the environmental impact and to boost the production. One of the main intention is the collaboration. This is not that is missing. We have a great collaboration between Albania and Kosovo, not only on the research, but also on the teaching process. A lot of academic staff and students are exchanged between countries and institutions, and we have been partnered in different EU-funded projects such as Erasmus capacity building. But we will not stop with this. We will go ahead, and the main collaboration on this field will be, let's say, on the shared breed conservation in order to better preserve endangered breeds, to have joint hatchery broodstock for having healthy population and to avoid inbreeding risk. Maybe to have regional brands such as, for example, Balkan Mountain cheese, and to have a collaboration through the research project. The emphasis will be in innovation by the use of new tools such as SNP genotyping, but as Professor Calucci mentioned that in the science there are no borders. Now the development of informatics, bioinformatics, artificial intelligence will help to see how not only to detect the SNPs, but also to see how the SNPs have impact on the structure, function, and behavior of molecules such as proteins or enzyme. Why not in the future to go with gene editing? Because up to now, there is in both countries no gene editing, no GMO, climate smart recirculating aquaculture system, and integration of renewable energy solution into biotech farming system. The final overview and vision is harnessing biotechnology to drive rural prosperity, preserving of native breeds, because are one of the sources for ensuring food security and also part of cultural heritage, building infrastructure through ISO accredited labs, fostering EU integration, and also making Albania and Kosovo as model for regional cooperation and bio-innovation in Western Balkans. Thank you.
Do we have any questions? No. Okay, so thanks a lot, Professor Hodan. And now we proceed with Professor Bashkim Ziberi from University of Tetova of North Macedonia. I'm going to wait a few minutes here next to me because I'm going to read a short bio of yours, which is really not short, but I'll try to shorten that. So Professor Ziberi finished his physics bachelor studies in Tirana, Albania. And then he did the MSc and PhD studies at the University of Leipzig in Germany in the field of condensed matter physics. He worked in a project financed by the European Space Agency, industrial projects and DFH projects in the frame of the research group 4845, and from 2007-2010 as a principal investigator working on surface engineering and guided nanostructuring of different materials using low-energy ion beams. He was the head of physics department and director of the Office for Scientific Research and Innovation at the University of Tetova. Dr. Ziberi, a scientific visit at ESRF European Synchrotron Radiation Facility as a beamline user. He is awarded for his scientific work from the European Materials Research Society. His member, a reviewer in a number of scientific journals, has more than 40 publications and over 100 contributions in international conferences, from which more than 20 are invited. Dr. Ziberi, from 2010, is board member of the Institute Alp Science that's aiming to boost the cooperation between the scientific diaspora with the scientists in the region. From 2018, he is member of the NanoAlp group established in the Nanotechnology Center in Tirana, Albania. He is also a Fulbright Scholar alumni from Dana-Farber Cancer Institute, Harvard Medical School in Boston, working on combination of radiotherapy and immunotherapy using smart biomaterials with nanoparticles for targeted treatment of cancer. He is a board member of the Global Oncology University and of the Program Committee Research Infrastructure and Nanobiomaterials at Horizon 2020 in Brussels, etc., and many others. So I'm not going to take you much time, but I want to listen to the presentation of Professor Ziberi titled Nanoparticle-Aided Radiotherapy Using Immunogenic Biomaterials in Cancer Treatments. The floor is yours.
So, hi everyone. Sorry, Ariola just last evening asked for me a short bio, so I didn't know what to send. I thought she will shorten it. Sorry for bothering you with that. So let me see. I will speak now. Let's go a little bit smaller in size from the previous talk and see how we can use nanoparticles for the combination of radiotherapy and immunotherapy for cancer treatment. As we know, in many countries, cancer is the #1 death cause, and it's increasing from year to year. And the challenges for finding, let's say, an appropriate method for cancer treatment is growing in different research groups. When Ariola invited me also, I wanted just to mention, thank you very much, Ariola, that I'm part also of the Nano-Balkan or Nano-Alp research section of the Academy of Sciences of Albania, and I'm working as a professor at the University of Tetova. Let me see. Yep. So most of us know that we usually use inert fiducials to trace the cancer sites in different organs in the human body. But we go now one step further. Instead of using only these inert radiotherapy biomaterials, how we can functionalize and use these materials also for other purposes. And that is, for example, when we load these polymer tubes with nanoparticles, and we can directly inject them in the tumor site. And through the controlled release of the biopolymers, we can release the nanoparticles. Why we do that? Because usually there are 4 main methods for cancer treatment: surgery, chemotherapy, radiotherapy, and immunotherapy. More than 50% of patients do radiotherapy, but usually you do externally, then you damage also the healthy organs. And so the idea is, how can you generate an intense enough radiation on the tumor site without affecting the healthy tissues of the human body? And the process, how the process it works, here are the antigen-presenting cells, because usually in the tumor site, we all know that the tumor cells are growing and growing and growing, never stop growing, opposite to the healthy cells that do the function and then die. But all these tumor cells, of course, they are kind of taking in prison the healthy cells, the T cells, the ones that are fighting the cancer cells. And what we do with the nanoparticles is that simply we generate enough radiation in the tumor cells that we make, so to say, the tumor bleed, and it releases the T cells. Once these T cells or antigen-presenting cells now know the genetic code of the tumor, they can go all over the body and also even attack the metastatic sites on the human body. The physics behind that is an old one, and it's based on the photoelectric effect or Auger effect. And it's schematically shown here. When you bombard with the atom with an outer electron, you can, if the electron has enough energy, you can knock out an inner part electron, and then you can generate this, you can make that one outer electron jump in this empty space here and an energy level, and then you generate a high energy photon because it's on the X-ray region. Or you can use the Auger electron that is sometimes when you, let's say, knock out the outer electrons. Now, the experimental part of all this, we have these tubes. These are biopolymer PLGA biopolymer tubes, biodegradable in the human body. You can fill them with gold nanoparticles, but with other high Z nanoparticles. And you can even combine them with immunogenic materials and release at the same time gold nanoparticles and immunotherapeutics on the tumor site. This is the release time of the biopolymer. The biodegradation of the biopolymer, you can control this biodegradation by, for example, using the pH value of the biopolymer and also the form how the biopolymer was generated, or you can even use different types of biopolymers. These are the mice CT studies, and we can observe here the SRBs that are implanted. You usually take the mice, you inject the tumors cells on both flanks of the mice, and you let them grow, and then start the treatment procedure. At one side, this is the treated side, this is the non-treated side. And as we will see later, both in the treated and the non-treated side, we have the effect of the reduction of the tumor volume. This is the SRRP, Small Animal Research Radiation Facility, where you can irradiate the mice and also do the CT of the mice. Gold nanoparticles in the prostate cancer, most of the experiments are here for prostate and pancreatic cancer. So the one is with no gold nanoparticles. This is with gold nanoparticles. And simply when you add gold nanoparticle to the in vitro studies of the tumor cells, you don't see any huge difference. But once you radiate the gold nanoparticles, we see the difference. That means you generate intense local X-ray radiation, and you cause, let's say, cell death. This is also for pancreatic cancer and prostate cancer, both of them, to show the abscopal effect. That means the also untreated site, it's affected by the radiation of the nanoparticles in the treated site, because once you release these antigen-presenting cells, the T cells, they travel all over the body and they are hunting for these cancer cells. And this is the Anchorage independent colony forming assay, so showing you the control group, gold nanoparticles, radiation only, and radiation combined with gold nanoparticles. That means once you activate these gold nanoparticles with photo effect or the Auger effect, you can cause the cell death. And if we want to study furthermore this histological analysis for the CD8 and let's say anti-cancer cells, And natural killer cell, we can observe that when we combine them with gold nanoparticles, the natural killer cell number is increasing much more than in case when we have only immunotherapeutic materials with gold nanoparticles without radiation and gold nanoparticles by itself. And also the antigen presenting cell number, it increases with activating high Z nanoparticles. The same is here also for treated and untreated tumors, so that we can observe that in both cases, for the treated tumor and for the untreated tumor, we have cell death. And if we see the CT images here, we will observe almost complete vanishing of not only the treated part, but also of the untreated part. That means it has also the metastatic effect when we, let's say, release the antigen-presenting cells in the human body. This is another experiment that we try to do to observe, do we need to irradiate the whole tumor, only on the subvolume of the tumor? And we show that it's enough to irradiate the subvolume of the tumor to effectively, let's say, decrease the tumor volume. And I will skip this one. And these are then the survival curves. And also for the treated and untreated part, we observe that in all cases, we don't need to use a higher dose radiation in order to have tumor volume decrease compared to higher doses. So we at around 5 gray is the optimum doses for having a higher survival rate in in mice in this case. Now, this, for example, is the case only for gold. Of course, nanoparticles, they don't have the effect only of the treatment now, but also have the effect of the diagnostics, because if you use the high Z nanoparticles, gold nanoparticles, or gadolinium nanoparticles, they are good traces because you can make them visible with MRI, CT, and different spectroscopic techniques, so that you can combine the diagnostic tools with therapeutic properties of these nanoparticles. And here only for gold are the different shapes, forms of nanoparticles having symmetric shape or non-symmetric shape. So this is the case with the gadolinium nanoparticles, as we can with the phantom, and in the mice in vivo, where we can observe the concentration of gadolinium nanoparticles and the release over time in the phantom and also in the mice. These are the three groups: the control group, the group that is only with, let's say, directly injected gadolinium nanoparticles. Of course, the directly injected gadolinium nanoparticles will not have higher concentrations because they will spread all over the organ. And if we inject these gadolinium nanoparticles merged inside the biopolymer, we can still observe the concentration of gadolinium nanoparticles and study their release over time. In 4 different mice, we see a very similar curves in all these 4 mice. That shows that in 4 different cases we have, it depends on the biodegradation of the biopolymer. We can use different techniques. I will jump this. So the idea also is one step further. Instead of using this SRBs to inject subcutaneously or orthotopically in the human organ, we can use microneedles. So especially you can batch them in the skin and have the controlled release of all this payload that I showed to you over time, so that you can control the amount of the payload that you are injecting on the tumor sites. I will jump this. So the next level, in fact, the last thing that we are doing now with our colleagues, and I'm working with my colleagues at University of Pennsylvania, Pittsburgh, and UMass Lowell now for the moment. We are submitting a project there. It's called Quantum Theranostic, where we exactly use the let's say, the combination of treatment and imaging of nanoparticles, and especially the imaging part, where we can generate the famous entangled photons. So the idea is because now the entangled photons has gained a lot of, let's say, attention in the last years because they have this special property, at least I haven't found that somebody really explains that how it works. How it works, yes, but why it works that way is that you generate two photons and one goes in this direction, the other goes in the other direction, but both of them, they have kind of correlation or physical, let's say, not contact, but they feel each other. What happens to the one, the other knows, and you can measure the, let's say, analyze the properties of an object with a photon that never contacted the side. So this is what we are using now, and we're using this entangled 511 keV photon pairs, and we use nanoparticle to enhance the entangled photon generation, because the problem is that usually in 1 million photons you generate one entangled photon. Now we have found the possibility of increasing this number of entangled photons in a very large amount. The idea behind is to use this kind of, let's say, additional machine or benchtop to the existing Linac accelerators for radiation, so we don't change the environment of treatment of the patient, and we will use different type of detectors, from single photon detector to avalanche detectors. And we are working with the colleagues here. We already have submitted the paper at Red Journal, and you can read it on arXiv. We have put it on arXiv for, let's say, critical discussion among scientists. And this is the idea how we generate the entangled photon from the annihilation of electron-positron, creation of the positronium, and then we have these two photons that are from the same quantum state, go in the opposite directions, and still they keep the contact with each other. Furthermore, we can also study the microenvironment of the tumors using this entangled photon, and this is one simulation from Gen 4 and Monte Carlo simulations that shows that when you this entangled photon, for example, encounter different types of cells, they can have different type of intensity in the energy, showing that depending on the type of the cell, you can have different height in the intensity or different full width half maximum of the peaks of the intensity. So we are in the process of learning by ourselves in this huge project, because it's more than 40 million US dollars that we are applying now, and we have, let's say, first stage is already, let's say, approved by the US government agency, and we are applying now in the second stage, and promise is very high that we get this high-profile project. At the end, I want to thank you for your attention.
Thank you very much, Professor Ziberi. If there is any question? Yeah, good. Can you also, hi, boys.
Are you sure you can really differentiate in prostate cancer the tumor cells from the non-tumor cells? Is it really that specific that you can actually avoid killing the non-cancer cells? And is it really better to use your method than just surgery?
Yes, at least in animals, it has shown, because surgery is, let's say, confined when you don't have metastasis. Once you have metastasis with the surgery, it will be difficult to remove all the cancer cells. And from the experiments that I showed here, you can distinguish between normal cells and cancer cells because of their size, for example, that they have. Because the normal cells are smaller in size than the cancer cells.
You're using antibodies, right? You're using antibodies to different.
No, the antibodies, we use them additionally because we can do that also without antibodies. The antibodies, we use it because we want at the same time, let's say, increase the autoimmune system of the body. But it's not necessary to use the antibodies. We just wanted to show that we can combine the techniques I can use only gold nanoparticles and send them to the tumor site without antibodies.
Yeah, but then how do you get the gold particles to the tumor cells specifically? You need something specific to get to them.
Yes, that can be done. We have done this by injecting it directly to the tumor site in mice, but you can do this through nanoparticle functionalization because the rest system otherwise will throw away the nanoparticles, but we do that with functionalization of nanoparticles so that they can long live in the human body until they are attached to the tumor site.
One more question?
Thank you, Professor. It's a very, very interesting talk. Pierce Cameron, University of Malta. So my interest is why do you think that over here you are getting a metastatic effect, right? Because if you radiate directly through the skin, you are still killing the tumor. You should still release the T cells and you should still get your metastatic effect. So do you think this is because your method is delivering a lower dose? You mentioned 5 gray. How much does that compare to doses when you are giving it externally? And do you think that this is the difference? Or is the external dose also killing the T cells maybe or something of the sort? Do you know why you seem to be getting a better metastatic effect here?
Yeah, we really don't know that why. In fact, there are still studies going in this direction, but it seems that when you send a higher dose, in fact, you kind of, for a very short time, generate a very high doses of X-ray on the tumor site, but that is very, very short lasting. But I'm not sure about that.
Okay, thank you.
So thanks a lot, Professor Ziberi. Okay, now it's time to proceed with a lady, with Professor Anila Babameto Laku. She is the head of the Genetic Lab Service at Mother Teresa University Hospital Center and at Medical University of Tirana, Albania. Professor Laku has completed numerous postgraduate specializations in medical and molecular genetics in Israel, Italy, France, and Germany. She's organized and led the work to establish 2 new laboratories in her service, the Molecular Cytogenetics Laboratory for the diagnosis and treatment of malignant hemopathies, and the Molecular Biology Laboratory applying modern sequencing methods for the molecular diagnosis of the most common genetic diseases in the Albanian population. She's working intensively on the application of NGS techniques for the diagnosis of rare genetic diseases and on completing the registry of rare genetic diseases according to the European platform. She's member of the European Society of Human Genetics and European Society of Cytogenetics. She is a participant and coordinator of several international scientific research projects, and she's made scientific contributions to the first genetic studies on chromosomal syndromes, the most common mutations in cystic fibrosis, beta thalassemia, Duchenne muscular dystrophy, and rare genetic diseases in the Albanian population. So the floor is yours, Professor. Okay.
I would like to thank Professor Batsu for the kind invitation to participate in this important event and also to present my speech, Genomics in the Diagnosis and Treatment of Rare Diseases in Albania, Current Status and Upcoming Challenges. Rare diseases represent a significant public health burden, and according to the European definition, a disease is considered rare where it affects fewer than 1 in 2,000 people. To date, about 10,000 rare diseases are known. It is estimated that the majority have a genetic etiology and 70% have a childhood onset. Their diagnosis poses challenges due to clinical complexity, limited access to advanced genetic testing, and few therapeutic options. The diagnosis process is frequently prolonged, expensive, and requires multiple genetic tests. Whole exome sequencing and whole genome sequencing, performed using a next-generation sequencing platform, have transformed the approach to these disorders. Our data on rare diseases represent the whole country because the Service of Clinical Genetics Lab is part of the University of Medicine and University Hospital Center Mother Teresa, where all rare diseases are diagnosed and treated. Our Service of Clinical Genetics Lab includes 4 units. Clinical Genetics Unit, where clinical evaluation, medical familial history, and pre-test and post-test counseling are undertaken by a clinical geneticist. Lab of Cytogenetics, with postnatal diagnosis for detection of chromosomal aberrations. Lab of Molecular Cytogenetic, that consists in genetics of hemato-oncological diseases, and Lab of Molecular Biology for molecular diagnosis of most prevalent rare diseases in Albania. Traditional diagnostic approach typically involves genetic tests such as microarray, single gene testing, and targeted gene panels. And microarray detect disease-causing copy number variants, including variants linked with microdeletion or microduplication syndromes. From American College of Medical Genetics was recommended on 2010 as the first-line microarray test for evaluating patients with development delay, intellectual disability, and/or congenital anomalies, replacing traditional karyotyping. Providing better care for our patient has required cross-border collaboration for microarray analysis. The table shows microarray results from 2012 onwards, obtained through international research projects or private laboratories. And so genomic syndromes such as DiGeorge, Angelman, Williams, Waardenburg type 4, Phelan-McDermid, and the rare structural abnormalities like 4p deletion are detected. In our lab, the MLPA technique is used to detect the most common rare genetic syndromes with microdeletion and microduplications. For single gene disorders with distinct phenotypes, Sanger sequencing provides high diagnostic accuracy. And even in these cases, performing the test on our patients required international collaboration, and some cases have already been published. So the case of alpha-sarcoglycanopathy or the case of mucopolysaccharidosis type 6. From our international research projects, over the years, we studied the most common genetic disorders in Albanian population and found that for cystic fibrosis, F508 deletion was the most prevalent mutation. Its frequency in Albanian population is 70 to 80%, and it is the highest reported in the Southern Europe. Regarding beta thalassemia, from the research projects, we have detected that 13 beta thalassemia mutations are identified, with 5 common point mutations, according for nearly 92% of mutations. Knowledge of prevalent beta globin and SFTR mutations in our population allowed development of a simple, reliable, and cost-effective genetic approach in our lab. Based on mutation prevalence, single base extension method is used as the first test, reaching a high detection. We apply target molecular techniques such as snapshot for beta thalassemia and cystic fibrosis, and MLPA for muscular dystrophy Duchenne and common genomic syndromes. Here are presented our patients and the panel of 8 most common beta thalassemia mutation. And also the panel includes the mutation for diagnosis sickle cell anemia. Also, for the cystic fibrosis, we use the panel of 11 most common mutation in Albanian population and intron 8 polymorphism variants to diagnose cystic fibrosis. Regarding the cystic fibrosis, Sanger DNA sequencing, or NGS, is recommended if only one or no casual mutations are detected. Understanding cystic fibrosis mutation classes in our patients is the first step toward personalized therapy for cystic fibrosis. Also, most patients can benefit from new modulator treatment, giving hope for the future, and implementing newborn screening for cystic fibrosis would improve outcome through early diagnosis. Regarding the Duchenne muscular dystrophy, most mutations are deletions, and in our practice, MLPA represents a reliable technique for the detection of small copy number variants, including single gene deletions. All the deletion duplications are provided by checking the reading frame on Leiden Open Variation Database. And genetic test results in this pathology are interpreted with clinical findings and family history and are essential for the personalized therapy. Here are the results of our patients with Duchenne muscular dystrophy, and the most frequent deletions range from exon 45 to exon 54. Most genetic diseases are not caused by the single gene mutation, nor do they have a recognizable clinical presentation. Therefore, NGS is rapidly replacing traditional methods in diagnosis of rare genetic diseases. NGS technology allows the rapid sequencing of millions of DNA fragments at the same time, and the whole exome captures only the coding region and is becoming a first-tier diagnostic test for rare genetic disorders, while whole genome also captures the non-coding regions, providing more data than a whole exome or other methods. The two methods range, the diagnostic yield ranges from 25 to 50%. Targeting sequencing is used for individuals with clinical presentations highly suggestive of a specific disorder. And the American College of Medical Genetics recommends NGS be used as a first line in patients with one or more congenital anomalies prior to 1 year of age, or intellectual disabilities development delay prior to 18 years of age. NGS show higher diagnostic yield in individuals with disorders affecting hearing, vision, musculoskeletal, skeletal, cardiovascular system, and multiple congenital anomalies. Classic trio sequencing increases diagnostic yield by 15% through detection of de novo and compound heterozygous variants. NGS analysis for our patient is not performed in-house. Samples are sent abroad through international research collaborations or private labs. We have recommended targeted testing in patients with clinical presentations highly suggestive of a specific disorder. So, like the case of Setter-Scholzen syndrome that has performed the analysis in Maggie's lab and is published from Professor Bertelli, Noonan syndrome, Treacher-Collins syndrome, or pachydermoperiostosis. Here are some cases of whole exome sequencing diagnosed, presented at European scientific conferences, like 3 families with KBG syndrome, or the case with Phelan-McDermid in 2 siblings in Albanian family. Whole genome sequencing enables the diagnosis of many rare diseases missed by whole exome, particularly those caused by larger CMVs, repeat expansions, or mutations in encoded regions. Here are presented the cases diagnosed only through whole genome sequencing, despite having normal whole exome results, like Waardenburg syndrome or Freeman-Sheldon. NGS variants are classified in two classes according to the guidelines, and the role of variants in the patient diagnosis is based on the presence in databases, comparison of patient's phenotype with known gene variant association, and screening for the variant in healthy and affected family members. Variants of unknown significance are the most frequent type or variant reported with NGS. And in some cases, also presented at European and Balkan conferences, the phenotype was consistent with no gene variant association, supporting the relevance of uncertain significance variants. Here we have the cases with spondylometaphyseal dysplasia or Kabuki type 2 syndrome. In cases where secondary findings are defined in our patients, or families and patients are informed about the potential risk and benefits unrelated to the patient's primary phenotype. American College of Medical Genetics has published a list of 78 genes and recommended reporting mutations in these genes. Reporting secondary findings enables early diagnosis, improves disease prediction, and supports prevention through timely medical intervention. Medical management changes occur in 30% of patients who obtain a diagnosis with NGS. Such changes include specific treatments, as in our cases, surveillance for additional manifestation, clinical trial, and accurate reproductive risk assessment. All of our diagnosed cases are recorded in the Rare Genetic Diseases Registry, maintained by the Clinical Genetics Laboratory Service. The registry is continually to be updated through interdisciplinary collaboration with medical specialties involved in diagnosis and management of rare genetic conditions. These data represent the first comprehensive genomic database of rare genetic diseases in Albania. In cohort of 206 patients, the distribution of diagnosed diseases is presented according to diagnostic methods, and rare diseases diagnosed by NGS are classified in monogenic diseases and genomic syndromes. And patients with a confirmed molecular diagnosis are grouped according to the primarily affected organ system. Neurodevelopment disorders are the most common, in 50%, followed by neuromuscular and metabolic diseases. Less frequent are skeletal dysplasia, connective tissue disorders, and other conditions. And in neurodevelopment diseases, monogenic disorders account of approximately 40% of cases, while 8% are caused by CNVs. De novo monogenic variants are well recognized as of neurodevelopment disorders as a cause, and 3 whole exome sequencing has a high diagnostic yield for identifying de novo monogenic mutations. Here are our results for the inheritance patterns of the diseases and also the variants pathogenic, likely pathogenic, and uncertain significance. Also, genetic testing in our in oncohematology follows a structured algorithm of genetic tests, and the testing strategy include fluorescence in situ hybridization and the next generation sequencing. In our lab is applied the method of fluorescent in situ hybridization, and also our pilot project for West development is enhancement of the molecular biology laboratory infrastructure in our service through the application of advanced genomic techniques. As a first step, we are implementing targeted NGN panels in oncohematology for myeloid and lymphoid malignancies, as well as rare solid tumors in pediatric patients. Despite progress, key challenges remain, including limited in-country access to high-throughput sequencing technologies such as NGS, insufficient funding for the routine implementation of advanced genetic testing, lack of sufficient manpower and trained professionals, and lack of national screening programs for early detection. Future efforts will focus on developing tailored diagnostic tools, improving infrastructure for local genomic testing, implementing neonatal screening programs, and integrating new treatment strategies such as gene therapy and personalized medicine. Strengthening genome medicine infrastructure and fostering international collaboration will be critical to overcoming existing barriers and improving outcomes for patients with rare diseases in Albania. Thank you.
Thank you very
I have
much,
a question.
Professor. There is a question for you.
So you've given us a very nice presentation of a very impressive setup. I have, however, a question. Did you discover in Albania, from the Albanian population, any new mutations of genetic diseases? New, novel.
Some novel mutation in rare genomic phenomenon. For example, like in Kabuki.
But these are variations, right? Right, but I'm talking like a new,
New mutation.
no,
No.
okay, thank you.
Okay, thank you, Professor. Okay, so we proceed to the next speaker. He is Professor Burim Ametaj from the University of Alberta, Canada. Professor Ametaj is working at the Department of Agricultural, Food and Nutritional Science at the University of Alberta, where he has established himself as a leading authority in nutritional immunology and dietary cattle health. With a PhD in nutritional physiology and immunobiology from Iowa State University and postdoctoral training at Iowa, Purdue, and Cornell universities, Dr. Ametaj has authored over 120 peer-reviewed publications and pioneered the application of metabolomics to understand and predict periparturient diseases in dairy cattle. His groundbreaking research has led to the development of novel biomarkers for early disease detection, innovative probiotic treatments of uterine infections, and new grain processing technologies to prevent periparturient disorders. As a recognized international speaker who has presented conferences across North America, Europe, Asia, and South America, Dr. Ametaj continues to advance our understanding of the complex relationships between nutrition, immunity, and animal health through cutting-edge systems biology approaches. The floor is yours, Professor.
Thank you. Greetings, everybody. I would like to thank the scientific committee, all distinguished attendees here, the Professor Ginushi, the President of the Academy of Sciences, and all the distinguished guests. So my presentation today is about a success story, how we started the idea of using probiotics to prevent diseases and also to treat them. So the world is in big trouble. Why? Because probiotics production in the world is so much that it can cover the world in 1 meter height. So, and there is a tremendous increase in the number of bacteria that are resistant to antibiotics. So we have to start looking for new alternatives. And this is why the title, Mining Probiotic Solutions as Alternatives to Antibiotics, from bench to market. So this is a story that how it started and how we are able now to sell this product in US, because US has more relaxed requirements for accepting probiotics. In Canada, for example, where I'm working, they do not accept this very much because they call probiotics drugs. And they are not drugs, they are just bacteria that we eat with yogurt on a daily basis. So the main issue that we have is that 30 to 40% of dairy cows are culled, removed from dairy herds every year. So there are more than 1 billion dairy cows. 40% means 400 million cows are culled every year for disease issues. But the main issue is that cows have infertility, and infertility is the main issue. The main reason for, number one reason for culling cows. So uterine infections causes infertility. And we use antibiotics, and there is antibiotic resistance. So this is what I'm going to discuss today. From identifying these probiotics from the vaginal mucus of dairy cows, a group of lactobacilli that we characterized. We did genome sequencing. We grew them in the lab, produced a product in a factory in the United States, in Iowa, and we now are we commercialize it and we are bringing it to the world. So 40 to 50% of dairy cows are affected by uterine infections, and they are associated with infertility. So we are this probiotics in different, in the North America and Europe are used a lot, and so this is the main reason for infertility. So this use of antibiotics is associated with resistance to of bacteria, and now we have bacteria that are resistant to multiple antibiotics, not only one, but up to 3 antibiotics. So we're going to run out of antibiotics, and if we don't find alternatives, we're going to be in trouble, because this bacteria, the genes that are resistant to antibiotics, they can transfer to humans, and then the antibiotics are not going to work. So this is our journey here. We started in 2005, the first idea. Nobody trusted us. Nobody could fund this, our idea to use probiotics to prevent uterine infections or treat uterine infections in dairy cows. So in 2008, we got the first funding. And then in 2010, the second funding, and 2016, the third funding. So in 2015, we started working. We established a company, a startup company, and started working on commercialization. We had the data to go ahead with this. So these are the 3 studies that we conducted. We tested the doses, how many doses we can give. So we started giving doses before parturition, before giving birth to the cows, intravaginally. So this was a new way of giving probiotics. Until then, probiotics were given only orally. This is the first time that was used, even in human medicine, has not been used intravaginally at this time. So we identified the probiotics. We tested them in vitro. We found that they all, these are the probiotics here, Pediococcus and Lactobacillus sakei. So we identified that they can produce pediocin by looking at their genes. So this was the antimicrobial peptides that they can secrete, and they kill the bacteria. So these are inhibitory zones here, showing that probiotics are very efficient in killing pathogenic bacteria, inhibiting them from growing. So these are the teams that worked, so we can switch that. So the first study, what we can get from this is that look at this vaginal discharges, which means that the cows have pus coming out of the vagina. 46% in controls, 13% in the treated cows. And then we had also, not only that, but we also had haptoglobin, which is an indicator of inflammation in the body, was lower in the treated cows. And then we had 3 kilograms per day milk produced from those cows just by treating them with the probiotics. So this is amazing. Even growth hormones that are used today in US, not in Europe, cannot increase milk production as much as we could with using probiotics. So this is the second study. We use different doses of lactobacilli, 3 doses here, 2 doses here, and this is the controls. So what we got, the same thing. We were able to lower the inflammation as indicated by lipopolysaccharide binding protein, which is a protein that binds bacterial endotoxin. Bacteria are covered by LPS, lipopolysaccharide. So this, we lowered it. We lowered also another indicator of inflammation, which is serum amyloid A. And also we were able to lower haptoglobin again, and we increased secretory immunoglobulin A in the mucus of the vaginal tract of dairy cows, so, which is amazing. By using probiotics, this shows the mechanism how they work. So they increase the immunoglobulins in the vaginal tract. We also lowered the metritis, the incidence of metritis in the infection of the uterus by control group 38 and the treatment 15%. And total uterine infections, including metritis, endometritis, inflammation of different layers of the uterus, and also pyometra, which is pus in the uterus inside. So we were able to lower from 53% in controls to 24 and 22 in the two groups that were treated with 2 or 3 doses of lactobacilli. So the advantage was 54.7 and 58.5% decrease in the incidence of these uterine infections. We looked also at the hormones, and we don't need to go through the details because the audience is so diverse and you probably don't understand all of these details. But you know, what happened was we improved in the blood, we improved increased progesterone, which is a female hormone that regulates reproduction, and also prostaglandin F2 alpha, which causes luteolysis of corpus luteum in cows. So this study here, we looked at the milk quality just by infusing these probiotics in the vaginal tract of dairy cows. In the milk, we had increase in immunoglobulin G compared to the controls. Also, haptoglobin in the milk was lower, so indicating that even so probiotics infused in the vaginal tract, they can improve mammary gland also health status. We were able to lower this haptoglobin in the milk, indicator of inflammation, mammary amyloid A, another indicator of inflammation, they all decreased. And then we increased also in this study 4 to 6 liters per day. In the first one, we increased by 3 kiloliters, but now we increased to 4 to 6 liters per day. So just by using probiotics, instead of using antibiotics, which milk usually is discarded, you have to discard the milk because it has antibiotic presence there. Okay, so this was study 3. This is the team here that worked on that. We applied intravaginal probiotics in 4 different dairy farms. One of those was organic, and we had some amazing results in the organic farm. We treated cows 2 times before giving birth. So this is like prevention of uterine infections. This is the first time in the world tested, and we were able to prove that to prevent uterine infections from occurring in the first place. And then we treated them after parturition at weeks 3 and 4. That could help treatment now. So prevention and treatment at the same time. And okay, so this is not okay. So again, here the main results that we see here is that uterine infections decreased from 50 to 29%. Milk also increased 4.6 liters per day in this study. And also we had an amazing decrease in the incidence of mastitis and milk fever. Milk fever has been with us in dairy cows for 200 years. And nobody knows exactly what milk fever is. So just by using probiotics, we lowered milk fever in organic dairy farm by 50%, 50, which is an amazing result because there's no ways how to prevent milk fever too. And the cows might die from milk fever. So this also study indicates there was increase in the secretory immunoglobulin A in the vaginal mucus of dairy cows. We lowered the systemic inflammation by decreasing lipopolysaccharide binding protein in blood. It decreases serum amyloid A. They are all related lipopolysaccharide. They bind lipopolysaccharide, which is endotoxin coming from gram-negative bacteria. And also we decreased the uterine infections by 30 to 50%. We expedited involution of the uterus by 21 days. And this is very important because if the uterus is not involuted, then you cannot inseminate cows to remain pregnant. And then we also increase prostaglandin F2 alpha, which, as I said before, it is the main luteolytic factor that luteinizes corpus luteum and makes cows available to be inseminated and remain pregnant. And then we improved also the metabolic status. We can continue this. So these are our publications on this area. And so now the other thing that we did is that we took this probiotic to commercialize it. And we created a startup company, Healthy Cow Corporation. And now we are producing the probiotic in Iowa, in Des Moines, Iowa, where is this company there. This is a factory plant that produces ProBioFerm, which produces probiotic from us. This is the package that can be used by producers. So you have cartridges here with probiotics, 300 mils. You can treat 30 cows with that. There is a gun to be used. So this is, again, the package for dairy producers. Come on. So this is a veterinarian here treating the cows. And we have also, again, here how it is treated. This is a dairy producer, Steen Farm in New York, which 1,000 dairy cows. She is very happy. There are 3 YouTube videos where she expresses her happiness with using these probiotics and the great results that they are getting. She continues to use to treat these cows. But one thing that I want to say, she is indicating here that the veterinarian had to eat his words and had. Why? Because he was doubtful that probiotics would have any beneficial health effects. So he accepted himself. He said now that they check cows, control, treated, control, treated, control, treated. He said, I have to eat my words in my head, because he has suggested to her, advised her to not use probiotics in dairy cows, but he was wrong. And here is another dairy producer, Jeff Nunn in Canada. He was very open-minded. He even invested in $200,000 to develop this product from his dairy farm income. And so this is the company that we established. I'm here. We have lawyers here that protected this technology. We have all these business people. This is Edward from US, a very well-known veterinarian there. And I'm concluding here by thanking General Dairy that supported my travel here, which I am the editor-in-chief. And so this is a very now impactful journal. In 2025, 3.1 impact factor. In 5 years, we brought this new journal to success. And with that, I'm concluding this presentation. Thank you very much, everybody, for being here. And if you have any questions, I am happy to entertain those. Thank you.
So I have a couple of questions.
Yes.
So it's not clear to me that you are following which are the molecular entities that are actually active in all these observations that you have. Do you know which molecules are actively helping you replace the antibiotics?
Oh, molecules I mentioned at the beginning. What are the molecules, the molecular entities that help in replacing these antibiotics? They are antimicrobial peptides, one, that these probiotics produce. One of those we identified, pediocin. And now we are working to apply this probiotic in other areas. I cannot talk about that because it is intellectual properties. We have identified even more of those because we've done genetic sequencing of these probiotics. And the other thing is that they compete also with pathogenic bacteria. For niches, and they also increase immunoglobulin, stimulate the immunoglobulin production. And so that's one, there are the multiple mechanisms.
So I think you may want to explore in your probiotics to use microorganisms that have antibiofilm properties.
Oh, they have.
They have.
They have, absolutely. They have
So which are those molecules?
Oh, now we are working on that, as I said, because this was just one project, but we are working in other areas. And we have found that they can inhibit the growth of very, very tough bacteria, Streptococcus aureus, E. coli, and many, many more. We've studied 9 pathogenic bacteria that cause diseases in dairy cows, and they are very resilient. The mechanism, we have not entered into the mechanism in detail, but these are the antimicrobial peptides that they can produce, so that we have found those into. Another very interesting thing is that we took the fluid where the probiotic grow, postbiotic is called, and then we tested this postbiotics against pathogenic bacteria, and it's working also. Not only bacteria itself, but they release something in the, and we are going to now, we have plans to do peptidomics with mass spectrometry to identify those. There might be others, but we are not aware at this time. Sure, I would be happy to talk, but certainly we have expertise at the University of Alberta also, but I would be happy to collaborate with anybody that is willing to work in this area. Yes. One more question. Go ahead, please.
My question is relatively connected with the colleague, but you mentioned in your presentation just 2 probiotic bacteria, but they are recognized among bacteria, a lot of probiotic, so-called probiotic. What's the uniqueness or difference if you compare with the other?
So the question was, you had 2 probiotic bacteria, but we have 4, in fact. What are the differences with others? We have not studied all the lactic acid bacteria that are present. Everybody, there are multiple groups working, but we were the first in this area, and so we have identified these 4. We have tested those in vitro. What the others are doing, we are reading what the others are publishing, but there are so many to study. But this is a very good example of how you can get probiotics to replace antibiotics. So that's for sure.
Okay, thank you.
Thank you. Thank you so much.
I think we can continue with the other questions if you have during the coffee break. We're already late, so the organizing committee is proposing we stop here the session. We go for a coffee break for 15 minutes only, and then we're back with an